It's my pleasure to introduce Dr. Seow, Meow with an S. And his topic today is how does smooth muscle contract and why do we care? And Dr. Seow is actually a specialist in smooth muscle cell biology and lung physiology. His current research focuses on mechanical function, ultrastructure, and biochemistry of airway smooth muscle in health and disease. And there are two components to his current CIHR funded research program. One of them focuses on the basic mechanism of contraction in smooth muscle, while the other focuses on airway hyperresponsiveness related to dysfunction in airway smooth muscle seen in asthma. Other research interests include skeletal muscle mechanics, whole lung and airway mechanics, the ATPase cycle associated with the cross-bridge cycle, and mathematical modeling of muscle and lung function. So Dr. Seow, thank you very much for taking the time to present to us. Over to you.
Great. Thank you, Dr. Limo, for a nice introduction. So I'll be talking about smooth muscle contraction, which is a topic of basic science, but I'll try to make it as clinically relevant as possible. We know that there are two types of muscles, striated muscle and smooth muscle. Striated muscle includes skeletal muscle and cardiac muscle, whereas smooth muscle is found in hollow organs, except the heart, such as stomach, bladder, ureters, blood vessels, airways, etc. Now, for a long time, we believe that the mechanism of contraction of smooth muscle is similar to that of striated muscle. But later on, we found that this is only partially true. At the molecular level where force is generated, the two types of muscles share the same mechanism, whereas at the whole structure of the contractile apparatus, they differ. They're different. And that kind of confers some special properties to smooth muscle. So I will start my talk with a brief review of striated muscle. As I just mentioned, at the molecular level, they do share the same mechanism. So this is a depiction of a striated muscle cell. It's very long and thin. You know, if you look at the skeletal muscle, they go from tendon to tendon, so it can be very long. But also the diameter is about 100 microns, so it's very thin and long. So sometimes it's called fibers, muscle fibers. And within a muscle cell or fiber, it's packed with something called myofibril, which is made of countless basic contractile units we call sarcomeres. So there are many sarcomeres arranged neatly in parallel and also in longitudinally or in series, and that gives us this striated pattern when we look at it under electron microscope or sometimes even a light microscope. So I'm going to zoom in to the sarcomeres, the basic contractile unit. This is obviously a cartoon depiction of the sarcomeres. This is one sarcomere, and you know there's 3 of them in parallel and 2 of them in series, and you know, a total of 6 sarcomeres. Now you can see that the shortening of a sarcomere is due to interdigitation of 2 sets of filaments, the myosin filaments and the actin filaments. So the actin filaments is depicted here as this green lines here, which is anchored to this vertical line, which is the Z line. And myosin filament is this red orangey filament with this fuzzy head sticking out of the myosin backbone. And these fuzzy heads It's called cross bridges, and it interact with the actin filaments to cause this motion of sliding. So if we zoom in on a single cross bridge, this is a cross bridge, is the part of the myosin molecule, and the long tail of the myosin molecule is embedded in the backbone of the myosin filament, which is not depicted here. It's outside of the picture. It will lie parallel to the actin filaments here, which is depicted here. And you can see that a cross bridge goes through cycles, and it basically attached to actin filaments and goes through a conformational change. We call it a power stroke, and then detach, and then reattach again, and then go through another power stroke. And that's what causing the sliding of the myosin filaments relative to the actin filament. And each cycle consumes one ATP. So the sarcomere structure works well for skeletal muscle or cardiac muscle. However, due to the sarcomeric structural limitations, it has a very narrow, short length or working length range. And that is because, if you let the muscle shorten too much to a point that a myosin filament could bump into the Z line, or double overlap of the actin filament could occur, and that would interfere with the ability of the muscle to generate force. Now, this never happens under normal situation. In skeletal muscle, unless you break the bone, then the muscle is no longer protected by the skeleton. Then it would enter this phase of contraction, and that is pathological. And also on the descending limb of the force length curve, this curve basically depicts how much force the muscle can generate at what sarcomere length. And that force is determined by the amount of overlap between the myosin filaments and actin filaments. So if you stretch the muscle, then the amount of overlap between the myosin filament and actin filament decreases, and the ability to generate force also decreases. And then to a point where there's no longer any overlap, then there'd be no force generated. Again, the muscle never operating at this extreme length. They're only limited to a 10 to 20% length range change. Okay, so that works well for striated muscle. What about smooth muscle? So if we look at smooth muscle cells, this is single cells isolated from rabbit urinary bladder, fixed at different bladder volumes. So this is obtained from an empty bladder. And then this is a full bladder. And that's, somewhere between, the bladder volumes. Now you can see that the length change of smooth muscle is enormous. It's about 7 to 8 times. And this is physiological range, because our bladder goes through this cycle several times a day. Now, remember that the working length range of striated muscle is very narrow. So how does smooth muscle function over this huge length range? So for a long time, people, we know this since the 70s, but for some reason, people still believe that the working mechanism of smooth muscle and striated muscle is the same. But obviously, the skeletal muscle model, the sarcomeric model, cannot be just simply plug into smooth muscle and say, that's how it works. It cannot explain these huge working length range. So a long time ago, we proposed this model that explains how smooth muscle can work in such a long range. Of course, this model is supported by many evidence. Actually, the evidence comes before the model, but I present it in a reverse way. Just make it easier to understand and easier to present the content. So this is, we call it a length adaptation, the model. So basically, a smooth muscle cell adapted to, let's say, a reference length, LREF, which is an arbitrary length. It could be an in situ length. So when the muscle is sitting at this length for a long time, they adapt it to that length. They generate maximum force with the maximum overlap between myosin and actin filaments. Now, if you stretch the muscle, let's say in this case to twice the length, the model predicts that it will be more contractile units inserted into the contractile contractile units array, which is a long chain of contractile units. That would increase the number of contractile units in series, and then it can be reversed if it's adapted to a shorter length, and the contractile units will be deleted. So what is the supporting evidence for this model? So let's look at the prediction of this model first. So when you adapt the muscle to a longer length, first of all, there'll be more contractile units in series. And that means that the muscle can shorten more because each contractile unit shortens a certain amount, the same amount. And if they arrange in series, this would be accumulative. So overall, there'll be more shortening of the cell adapted at a longer length. And because each contractile units work independently, so they shorten a certain amount and over the same amount of time. So the amount of shortening over the same amount of time is increased. That means the shortened velocity of the muscle cell adapted at a longer length will be greater. And also the model predicts that the power output of the muscle will be increased if you adapt the muscle to a longer length. And that because there's more contractile units, they consume more energy, and therefore they have more power output. So the data here, we a long time ago, we found this, and that is the force velocity curve, which is this hyperbolic curve of a muscle adapted to a shorter length, compare that to a longer length, and the short ones in red. So the velocity you measured at different loads is consistently lower than the same muscle that's adapted to a longer length. So this muscle is able to shorten faster at the same load, but just simply adapt the muscle at a longer length. So velocity is increased, that's consistent with the model prediction. And the power also increased, consistent with this model. Okay, so the other prediction of this model is that there are more contractile units in the muscle cell adapted to a longer length. Therefore, we should find more myosin filaments in longer cells. And so is that true? So if smooth muscle is like striated muscle, that we can see the contractile units, these are the Z lines here, and in between, these are the myosin, the sarcomeres in parallel, and those are sarcomeres in series. So if we can see the contractile units in smooth muscle, then it's very simple. We basically adapt the muscle at a longer length, for example, and we fix the muscle and we look at it under the electron microscope and in the longitudinal section, and count the number of sarcomeres in series and see if it indeed increases with increasing cell length. However, if you look at smooth muscle, same magnification, but you hardly can see any obvious contractile units. And this is why smooth muscle is called smooth muscle, because it does not have this striated appearance. You can zoom in and see the myosin filaments and even actin filaments, but there's no way you can tell when the contractile unit starts and when it ends. So luckily, we can take a different look. Instead of looking at the longitudinal section, we take the cross section of the muscle, which looks like this. Now, you can see the myosin filaments. These bigger black dots here, these are the myosin filaments. And the little dots surrounding the big dot is the actin filaments. So in the cross section, the muscle cell cross section, we can count each of the myosin filaments and see how many myosin filaments we can find in the muscle cross section. So this kind of a measurement can help us verify whether the model we proposed is correct or not. So we start with the hypothetical situation or a thought experiment. So if we said the muscle cell has myosin filaments inside the cell, which is these red short lines, an assumption here made is that the number of myosin filaments is the same in the short cells and when you stretch the cell. And they are more or less evenly distributed. And we know that the volume of muscle cell is conserved at different lengths. So when you stretched a short cell to twice its length, the cross section would decrease by half, to conserve the cell volume. And therefore, if we take any cut of the cross section and count the number of myosin filaments, we'll find that short cell should have more filaments than the one in the long cell. Right? And to be exact, if the number of myosin filaments is the same, and if you stretch the muscle to twice the length, then you would expect the number of the myosin filaments to be half of that of the short length. So let's see if the measurements actually back up this thought experiment. So first we look at the force generated by the muscle at two different lengths, the arbitrary reference length and twice of that length. And the force generated or stress generated by the muscle at two different lengths, the same muscle at two different lengths, is about the same. So that sort of supports the model that, in adapting the muscle to different lengths, the isometric force generated by the muscle should be the same. Now, look at the cell cross-section. The long cell is almost exactly half of the short cell. So that confirms that the cell volume is conserved. Now, the last prediction is that number of myosin filaments per cell cross-section in the short cell, and the long cell, we expect it to be half of the long cell. However, it's significantly more than half. And that means that there are more myosin filaments generated or somehow appear in the muscle cell when you adapt it to a longer length. So again, that is the consistent with this model, that when you adapt the muscle to a longer length, it would have more myosin filaments. Now, this process of length adaptation does not happen instantly. It does not mean that when you stretch the cell to twice its length and suddenly you would have more myosin filaments and the muscle can, do different things. No, it takes about 5 to 20 minutes to this adaptation process. So I'm showing you some of the, I guess, illustration of how this adaptation occurs. So this is force generated by smooth muscle if you stimulate it. And this is the muscle adapted to a certain length. And then when you remove stimulation, it relaxes to the baseline. And if you oscillate the muscle, stretch and release, you can also do just a stretch, or you can just do a release, or you can do a repeat stretch and release a couple of times, and like a length oscillation. Then after that, the ability of the muscle to generate force is reduced. And then if you let it sit there and stimulate it every 5 minutes or so, then the force would recover and basically go back to this one. So this is the process of adaptation. Usually the first 5 minutes, it would recover by about 80%, and then the rest would happen over the next 10, 20 minutes. Now, this is the basically the plot of the active force, or the ability of the muscle to generate force, versus the amplitude of stretch. So the more you stretch and release, or amplitude of length oscillation. So basically is this, from this point drop to that point, after a certain oscillation with certain amplitude. So, and we found that this is more or less a linear relationship. The greater the amplitude of stretch, the greater the decrease of active muscle force. And of course, if you let it sit at this certain length and stop the length agitation, then the force would recover, and then they will, becomes the same as the force pre-oscillation. And this is another way of looking at it. So right after length oscillation, this is about 20% of length oscillation amplitude. So you can see that the active force, or the ability of the muscle to generate force, is decreased, and then it recovers over a period of time. And also over the same period of time, the myosin filament density in a muscle cell cross-section also follows a very similar time course. So suggesting that these two may be related, and that is that as the muscle recovers the ability to generate force, the more filaments are formed. And it should be the other way around. The more filament is formed, therefore the muscle is able to generate more force. So this is the so-called the process of length adaptation. So we can sort of summarize here that there are 3 steps in the length adaptation process. One is first is a mechanical trigger. It's either a stretch or release or a oscillation. And then it following this is the disassembly of contractile units, which is reflected in the decrease of the contractile force and the myosin filament mass. So that's this point right here. So right after oscillation, the muscle is not able to generate maximum force, and the myosin filament density decreased. So we take that as evidence that there is a disassembly of contractile units, dissolution of the myosin filaments. And then it's followed by reassembly of the contractile units, reflected in the recovery of contractile force and myosin filament mass. So it's like, a mechanical trigger is a signal for the cell say, oh, okay, we're going to operate at different lengths now. So first thing it does is it disassembles existing contractile apparatus. And then once the mechanical perturbation is removed, then the muscle cell start to reconstruct the contractile units and contractile apparatus so that it would fit the length, a new length of the muscle cell. So it would achieve optimum overlap between the myosin and actin filaments, and therefore generate more force. So now we are in the second half of our talk, and is why do we care about this unique property of smooth muscle? There are many reasons that we should care, but I'm just going to do two examples. And the first one is the shifting of the so-called length-force relationship. Now, the solid lines are the smooth muscle length-force relationship, and this dotted line here used as a reference is the striated muscle length-tension relationship, which is permanent because this relationship is governed by the structure of the sarcomere. So unless the structure of the sarcomere changes, this relationship would not change. It's a permanent, but smooth muscle is different. So here when you adapt the muscle to a longer length, it would have this sort of length tension relationship. I only depict the ascending limb of the length. There's a descending limb that we get ignored at this point. Now, if you adapt the muscle at different shorter lengths, the same muscle, just let it shorten it half and adapt it there, it would have a different length-force relationship. So there's something that happened inside the muscle cell that allows this shifting of the length tension curve to occur. And that it is what enables the muscle to operate at a long length, And this is basically predicted by the model that we proposed, that if you have more contractile units in series, you would have this relationship, and if you have less, then you would have this kind of relationship. So let me use an example to explain the physiological function of the shifting of the length tension relationship. So let's say the uterine smooth muscle contraction during childbirth. So at the beginning, the uterus is big and the muscle is stretched and sitting at a long length. And then during the childbirth, first, the childbirth process is not just one single contraction and that's it, but it's actually a waves of contractions and relaxation. So first, the muscle would contract, and that's assuming it contract against some load. Here, just arbitrarily, I say 50% of the maximum force the muscle can generate. So it starts up here, and it shortens. And when it shortens, the amount of overlap between myosin and filament and actin filament decreases, and therefore the ability of the muscle to generate force decreases as it shortens. And then when it's no longer greater than the load, it would stop. It would stop shortening here. Now the muscle relaxes, and then it recovered. Due to the length adaptation, it recovers somehow here, and let's say. And then the next wave of contraction comes, stimulus comes, and the muscle contracts, and now it can shorten more to this point, and then it will stop shortening. And again, if you let it sit there, it recovers, and then due to the adaptation process. And now if another stimulation wave comes, then it will shorten again, and it would just keep repeating this process, until the fetus is expelled from the womb. So, so this is one of the physiological significance of these smooth muscle properties, and that allows the muscle to work at a much longer length range than striated muscle can. Now, under pathological conditions, this could be a problem. It can be a problem. For example, let's say this is a arterial smooth muscle from a normotensive individual. And if for some reason, and when the muscle contracts, smooth muscle contracts, in the regulation of blood flow, it's limited the range. It's the shortening is self-limiting, and basically it would not shorten too much to cause problem. However, if for some reason, if we allow the muscle to adapt to an abnormally short length, then it can shorten much more, right? So the difference between here and there, it could be a difference between a normal tensive individual to a hypertensive individual, or a non-asthmatic to a asthmatic individual if this is airway smooth muscle. So for a long time, our strategy of treating hypertension and asthma is to reduce the contractile force of the smooth muscle. And however, with this new insight, adaptation or maladaptation should be considered as a possible mechanism that cause these diseases. So this is one of the areas that our research program is focusing for, is how the adaptation process cause pathology in different diseases. Now, the other reason that why we should care about how smooth muscle contract is found in this phenomenon we call response of asthmatic and non-asthmatic lungs to deep inspirations. So a deep inspiration, basically you take a big breath from the FRC, which is the functional residual volume, functional residual capacity to total lung capacity. And that, roughly doubles the volume of the lung. And if you take a few big breaths, it has this bronchodilatory effect, especially if the lung is challenged with bronchoconstrictors such as methacholine. So in this experiment, they have two groups of individuals, non-asthmatics and asthmatics. So before they, if they don't allow the test subject to take a big breath, so there's no deep inspiration, and if they give them a single dose of methacholine, their airway resistance would increase to this level. Usually it's down here somewhere. Now, in non-asthmatic individuals, if they allowed the subject to take 2 big breaths or deep inspirations, the resistance decrease. And if it's allowed to take 5 such big breaths, then it further decreases. And asthmatic individual doesn't seem to respond to this bronchodilatory effect of deep inspiration. And the reason we still don't know why, and actually this is one of the focus of my research program, is to figure out why there's a difference in response. And if we can figure out the reason, or even if we can make asthmatic airways to respond more like the normal ones, maybe we can have a better treatment for asthma. But we're not talking about asthma today. I'm just giving you the basic normal physiology in this talk. So this big breath. So what happened to, when you take a big breath? Basically, I say when your lung doubles the volume, everything inside the lung gets stretched, including the airways. So could the stretch of the airways that act as a mechanical trigger to cause this process of length adaptation? And therefore, that is why we see this bronchodilatory response. Now, this bronchodilatory response is temporary because if you stop taking big breath, then the airway resistance was slowly creeping back up because the stimulus is still there, the methacholine is still there. And this usually takes about 5-10 minutes for the bronchoconstriction to occur. Now, it turns out that in healthy, well, maybe even in asthmatics, it turns out that we take a big breath every 5 to 10 minutes. Even if either we know it or we don't, unaware of it. There's a study saying that, on average, we do take big breath once in a while. And that probably is the sort of the first line defense of bronchoconstriction or excessive bronchoconstriction. So, when we feel like we don't have enough, if we feel like we're short of breath or something, usually we take a big breath. Now, the question is, if this big breath, is it enough to stretch the smooth muscle inside the airways, and to what degree? And that is the question. Like this, I showed this before. If the stretch is 5% or less, then maybe the smooth muscle would not play an important role in the bronchodilatory response. But if the stretch is big enough, then we can see there's a significant reduction in the ability of the muscle to generate force. Although this decrease is temporary, because with adaptation we know it would readapt and then generate the same force again. But at least within a window of 5-10 minutes, you would have a temporary release of bronchoconstriction relief. So the question becomes, how much can an airway be stretched or distended during a deep inspiration? And then is this deep inspiration induced stretch sufficient to reduce bronchial smooth muscle contractility? Now, if we have answers to these questions, then we can assess whether this smooth muscle plays an important role in this bronchodilatory response of deep inspiration. So to address these questions, we designed some apparatus. These are homemade, and basically to house the isolated lung. This is a sheep lung, and basically we build the whole thing onto a plank, sitting on the plank, and the whole thing can be lifted up from the trolleys down here and placed on the stretcher of a CT scanner. And this chamber that houses the lung can be placed within the gantry of the CT scanner. And so we can measure lung function, like resistance, elastance, and lung volume, etc. While the lungs being scanned, where we can look at the diameter of the airways. So these are the airway trees from the CT scan. This is at FRC, the functional residual volume, and that is basically the base, the lung volume that we have at rest when we are not taking a big breath. And then this is at the higher lung volume, and this is at the TLC, total lung capacity. So we identify each of the airways here. So for example, this one here, which is that here and is that over there. So we can measure the diameter of these airways change during the deep inspiration. And what we found is this. So we have measured many airways, and we sort of arbitrarily divided them into 3 groups: small airways, medium airways, and large airways. So the large airways, it increase the diameter at this is lung volume here now. So at the lung volume of in sheep, actually, it's not, it's only 1.5, and then in humans, usually it's 2.0 here. So in all the size of airways increase when we take a big breath, and if we normalize this change to the volume, the diameter of the airways at the FRC, that's before we take a big breath. And what we see is that for large airways, the increase is about 20%, and then for small airways is over 60%. But on average, this black dots shows is the average of the total number of airways. On average, the increase is about 46%. So now we have answer to this question: how much an airways can be stretched? So we know it's roughly about 46% in sheep lungs. Now, is the deep inspiration induced stretch sufficient to reduce bronchial smooth muscle contractility? So to address this question, we dissect out the bronchial smooth muscle. Well, we dissect out the airways, and the same lung that was scanned by CT. So we know that that inside an intact lung, they expanded by 46% on average. So we isolated some of the airways and we cut them into airway bronchial rings, and we can stretch those rings to the same amount, and that is 46%. And then we see how much that affects the ability of the smooth muscle that is embedded in the bronchial ring, how much the contractility of the smooth muscle there is affected. And this is what we found. And so before we stretch the muscle, it generates a certain amount of force, a maximum force. And then in here, we actually do a 3 stretch, and by 46% on average. And we can see that there's a significant decrease, over 40% decrease in the ability of the muscle to generate force. And then, when the stretch stops, when this, the muscle sits in that static length without any perturbation, it recovers its ability to generate force over a period of time. So if we take a big breath every 5-10 minutes, that means our airway muscle in the lung is actually never operating in this range, but basically in this range. After a deep breath, it would go down here and then recovers over 5-10 minutes, and then another big breath would reduce it. And therefore, it limits the contractility of the airway muscle to a substantially lower force than it otherwise would be able to generate. And that is probably the mechanism of healthy non-asthmatic individuals, how they can basically respond to a deep inspiration and reducing the bronchoconstriction. Okay, so the take-home messages here. So length adaptation, which is a unique smooth muscle property, because that does not happen in skeletal muscle. is underlying the ability of smooth muscle to function over a large length range, by shifting the length tension curve that I described. And now the adaptation process includes disassembly of contractile apparatus upon a mechanical trigger. And then it's followed by a reassembly of contractile apparatus to fit in the new cell length, thus recovering maximum contractility. Now, deep inspiration significantly stretches the airways muscle within the airways to transiently cause disassembly of the contractile apparatus and the accompanying loss of contractile force, and thus bronchorelaxation. So basically, I have introduced a new concept, for people not in the field of smooth muscle, because in the concept of length adaptation is actually now relatively accepted because it appears in textbooks. For example, the Byrne and Levy physiology textbook that we use while I was in graduate school now has a chapter on this smooth muscle that includes length adaptation and stuff. So it's gradually being accepted, and so it's not new. But I think for people outside of this field, this is probably a relatively new thing that you're seeing it here. So with that, I thank you for your attention, and I'll be glad to answer questions.
Thanks very much, Dr. Seow. Very interesting. It reminds me of when I'm learning physiology, etc. So that was very a blast from the past for me. And yeah, it's interesting how far things have come. Any questions for Dr. Seow? I don't see anything in the chat.
Mm.
No, I think that's it.
Okay. Yeah, I think it's a basic science question, but has some relevance to pathology. But I think it's for people who are not in the field of asthma, it's probably a new concept, and hopefully they would learn something, and then when they encounter something, they could remember this behavior of smooth muscle, and that may explain some of the pathology that they saw.
Yeah, I've got a friend who's got asthma, so it's interesting for me for this. Obviously, if it translates into some sort of treatment paradigm, that would be great.
Right.
Okay. Thank you very much, Dr. Seow. Enjoy your weekend, and thank you for presenting to us.
No problem. Thank you. Bye.
Bye, everyone.